Campylobacter is a atypical pathogen. It's not like E. coli or Salmonella. It has a unique biology and survival mechanisms. The diversity of Campylobacter at pre-harvest on the farm is much higher than the diversity of Campylobacter on the end meat product.
Welcome to Unplucked, the official podcast of the Poultry Science Association. This is the podcast where we strip away the fluff and get real about poultry science from bench to barn. I'm your host, Andy Vance, Executive Director of the Poultry Science Association. Well, today we're joined by Dr. Issmat Kassem from the University of Georgia's Center for Food Safety to take a deep dive into one of the more challenging pathogens that we're studying in poultry production. With more than 20 years' experience and 100 publications to his credit, Dr. Kassem's research tackles foodborne pathogens and antimicrobial resistance using innovative, antibiotic-free approaches. In this week's episode, we're going to talk about his latest U.S. poultry-funded study on processing tolerant Campylobacter jejuni, a major food safety concern, and then what the industry can do to better detect and control these resilient strains of Campy. Dr. Kassem, let's get right to it. Would you start maybe with a quick overview of this recent project and what motivated you to focus on processing tolerant Campylobacter in the broiler stream?
Sure. Thank you for having me, Andy. I have been working on Campylobacter since 2009. Mainly, I was focusing on pre-harvest. I started at Ohio State University, and since then, I've been working on biology and ecology of Campylobacter as mainly related to poultry and how it causes human infections. During our work and the work of other colleagues that are involved in Campylobacter research, we always noted that the diversity of Campylobacter at pre-harvest on the farm is much higher than the diversity of Campylobacter on the end meat product. Okay, so the number of strains, their genetic diversity is much higher on the farm than on the actual end product. And this is because obviously and logically, the chicken meat goes processing to clean it up, right? So when you're cleaning up, you're killing some of the stuff, you are eliminating some of the stuff, but some of the Campylobacter remain. So based on that observation, that was the first observation. So there is more diverse Campylobacter on farm, less diverse Campylobacter on the chicken meat because of processing. And those that are persisting on the chicken meat, when there is an outbreak, those strains that are persisting, that remain after processing, appear to be associated with the problems that we hear about human infections and outbreaks and whatnot. So the question becomes is that those strains that stayed on the meat during processing, why did they stay there? Is there a somehow they are able of resisting or fighting back or whatever it is against the interventions that we are introducing during processing? So it started from there. Campylobacter is a atypical pathogen, if you want to think about it. It's not like E. coli or Salmonella. It has a unique biology and unique genetics, unique survival mechanisms. So putting all these together, we came up with the question, would the Campylobacter that remain on the chicken meat, are these capable of potentially tolerating whatever we are introducing to kill off Campylobacter during processing?
Looking at that piece of the puzzle there and what you were looking to address with this project, and pull back maybe for some of us who aren't as well steeped in processing and products as you are. So, for the uninitiated, how does this particular strain of Campylobacter typically end up on these products to start with, and what challenges do we face in trying to control this particular bug during the processing process?
This is a very good question. So Campylobacter loves chicken, loves broilers. So it lives inside the gut, and apparently, or largely, the relationship between Campylobacter and the chicken gut does not adversely affect the chicken necessarily. So the Campylobacter is there, of course, because the environment there is suitable for Campylobacter growth in the chicken gut. And there doesn't appear to be major and significant impact on the chicken itself. Of course, it has been associated with some diseases, but those diseases in chickens seem to vary according to chicken breed and to vary in severity. So the relationship has been regarded by many to be sort of commercialism. It's a part of the natural microbial community that lives in the chicken gut. And the chicken gut actually represents a very good place for Campylobacter to live. Chicken temperature is higher than the human body, it's around 41 degrees Celsius. And Campylobacter is thermotolerant. Campylobacter is microaerobic, it likes low oxygen conditions. So it's just an ideal place for the bacterium to live in there. And because it is an ideal place, and because there isn't a severe effect on the chicken, to put it this way, or significant effect on chicken health necessarily, the relationship seems to be ongoing and to be persisting. Because of this relationship, it becomes difficult to eliminate Campylobacter from chicken. For example, because it doesn't cause severe disease in chickens, it's not easy to devise vaccines against Campylobacter in chicken. And because it loves the environment inside the chicken gut, it will grow there and it will grow in abundance there. And this abundance, like for example, in the chicken cecum, which audience would be familiar with the cecum, which is part of the gastrointestinal tract of the chicken,
Mhm.
you can find up to 10 to the 8 sometimes, or even 10 to the 9 CFUs of Campylobacter. So it's abundant in the chicken gut.
Yeah, it's a lot of critters.
Yes. And if you take the chicken and you want to process it, of course, you want to eviscerate it, you want to cut out the guts and whatnot. But the cecum is a very sensitive organ, so it can be easily ruptured. And when you are rupturing the cecum, whatever inside the cecum is going to spill and cross contaminate the meat. So that abundance and that relationship makes it a sort of inevitable in a way to have Campylobacter in chicken. Now, the problem is Campylobacter is a human pathogen. So if we eat Campylobacter contaminated chicken, yeah, we can get sick. But the relationship is there. That abundance, that relationship between Campylobacter and chicken makes it hard to control Campylobacter on that final problem.
Looking at the specific strains you've identified that withstand some of the most common antimicrobial treatments, whether it's PA, chlorine, other things that we would typically find in a processing facility to address some of these pathogens, what is it about these strains that have made them more tolerant to those typical interventions than others?
This is a good question. Talking about Campylobacter genetically, when you look at the genome of Campylobacter, it's much more limited than compared to E. coli, for example, or Salmonella, right? So they have those limited genomes, but yet they have this ability to evolve and diversify, especially in an environment like a farm. So they have the ability to evolve, right? So they are evolving. These are pathogens that are evolving. Now, those strains that are persisting, they have specific genome content, the specific genes, specific DNA. We don't know precisely what makes them at this moment more tolerant. But what we know is that they are different. And we are trying to pinpoint which differences that contribute to this processing tolerance, right? And to do that, we have to go systematically. So you get the strain, you look at the genome of the strain, and you identify those differences, right? And then one by one, you try to tackle those differences and understand how they are contributing to that tolerance. We are in the step where we are trying to identify those different genetic markers that are involved, but we know for sure that they are different, genetically speaking.
So you've got different genetic markers, some real differences here. When you look at how these bacteria survive and maybe why they're more tolerant, what's the current thinking about what these differences tell us about the limitations of some of those current interventions we're using in processing and how we might have to adapt our strategy?
I work with other pathogens, Salmonella and E. coli, but my experience with Campylobacter is that you always need to think differently. Despite many, many years and a lot of research on this pathogen, on Campylobacter, its ecology and biology still remain. There's still a lot to learn about the ecology and biology of Campylobacter. So this uniqueness, like for example, one of the unique features about Campylobacter is the way it metabolizes energy. Okay, so like in the chicken gut, there is a molecule there that can serve several molecules, can serve as primary energy source for Campylobacter. So the way they metabolize energy, the ability of Campylobacter to use different substrates in the gut to metabolize energy and survive, that's one factor. So we refer to this as respiratory chain. It's highly branched, Campylobacter, allowing it to persist and generate energy in different environments. So even if it's in the gut, then it goes out of the gut. It can survive outside of the gut using this energy metabolism. The way it survives is also in a way unique. And this uniqueness can be translated into overexpression of certain genetic features or genetic pathways that can allow these strains of Campylobacter to persist. Changes, mutations to the cellular membrane, changes or expression of genes that can resist the antimicrobials. In addition to all of that, there is the ability of Campylobacter also to survive under viable but non-culturable stage, where it enters into a physiological stage where it becomes hard to detect under stress, right? And the VBNC state, for example, with Campylobacter, there's still a lot to understand about. So it's a combination of biology and ecology that allow these strains to persist. These are intrinsic property to the bacterium itself, but there are also characteristics of the hosts, which is the chicken, can also contribute. Again, we go back to this relationship between Campy and chicken that allows those strains to persist in this environment or host.
When you looked at some of the results of this particular project and what you've learned about Campy through your research, are there specific control measures or changes in processing protocols that your research suggests might be more effective, or things that you would recommend to industry at this stage?
I realized that challenges that are associated with introducing anything new
Yeah.
to the processing. And I should stress something that's really important. Poultry processing in the States is effective to reduce the pathogens. It's effective, it works, it's great. Okay.
Hmm.
What we are looking at is not change, is like how to make these better.
Sure.
What can we add to make them even better? They are good, but we want them to be better, right? So one of the things that I have been exploring, I have a couple of things. So on the level of processing itself, not to talk about prior to that, I'm exploring the use of antimicrobial light. And this is, we have tested this and it's really effective. Of course, it's in experimental stages, but the question would be, will it be really effective under the high traffic processing environment, right?
Right.
So there's antimicrobial light, and also we evaluated new phytochemicals that are safe to humans and do not impact the sensory qualities of the meat. And we identified a new phytochemical. And what we are doing with these is that we are not looking to replace what's being done. What we are hoping to have is add
Mhm.
to what's there to make it better. So if you have like a PAA, for example, and it's giving you X log reduction in Campylobacter, what if we add this light? Would we have an additive effect? Would we achieve more reduction, making the food even safer? We want to make it as safe as possible, right? We want to disrupt this relationship between Campylobacter and chicken that's allowing those Campylobacter to persist. And to do so, we have in a way to think a little bit out of the box and like a hurdle mechanism. And so you cannot just rely on one thing. You need different things. So if you have your traditional antimicrobials with these new interventions in the lab, actually, they have been working great. So that's what I'm looking at, something to add, something that can be easily integrated into the processing to allow for a safer product in general.
I'm glad you started with acknowledging, change is hard and we have a good system now. But I want to think about, if we don't take action, as you say, to kind of continue improving the system, to think about things we could add. What's the potential risk on the public health side of things if we let these tolerant strains of Campy go undetected or untreated? If we don't do anything, what's the potential risk?
We can look at what we have currently. And let's take a step back. So, when it comes to safety, if we're talking about the safety of meat, the safety of poultry meat is the job of everybody involved. It's the job of the producer, the processor, the supermarket, and also the consumer, right? What I believe we have been doing greatly so far, we have been reducing those risks. We have to maybe add a little bit of education for the consumer so that the consumer understands how to handle. As we said, Campylobacter is going to be on chicken meat, right? In many instances, you cannot eliminate it totally. You cannot get zero Campylobacter. But this education part, I think we need to do more. There has been lots of work on it, but we need more education on the level of the consumer. The system as it is, are there sicknesses out there associated with Campylobacter that has been traced back to poultry? Yes, there are. There has been, right? So this tells you, despite having a very good system, despite investing from the farm all the way to fork, right? there are still things that are going to happen. So if we want these things that are going to happen to reduce them even further, right? To build more confidence in a wonderful protein source for the nation, for the American citizen. Can we do something? Yes, we can. And we shouldn't be afraid. One of the things is, yes, you want to rely on your tried and proven
Sure.
approaches, but we shouldn't be afraid to look outside the box. So currently, we have some issues, right? I strongly believe that these issues, these like infections or outbreaks or illnesses, can be reduced further if we take the step to, okay, we have what we have, it's great, let's build on it, right? To have this decision. And I believe that this is a decision that's taken by the industry. It's just the matter of finding the right fit that's effective and economically feasible and all that good stuff. So it would be much better to build on what we have. The other thing is that I have learned after 25 years of being microbiologist, you shouldn't put all your eggs in the same basket, right? Okay, they are tried and tested, but if you are going to just rely on those, well, what if a bigger issue happens? So be preemptive in a way if you want. And if you want to look at it this way, being preemptive, let's find something that can make things even better. And we shouldn't be afraid of looking into it.
That's such a great message, Professor, and I really appreciate the way that you frame that. I think that's good advice for all of us, whether we're working in pathogen reduction or life in general. When you think about the next steps, one of the things I love about science and the research process is we learn something that means there's more for us to learn. So I'm curious for you, for your team, your lab, what are the next steps you're working on or thinking about to advance detection and control of Campy and these resistant strains in particular?
We're doing several projects. One of the projects we're working on is when it comes to detection, for example, isolation of Campylobacter takes a long time. Isolation and enumeration of Campylobacter. We're looking at ways to reduce that. So it can take up to 4 days between enrichment and isolation. And sometimes when you enrich, you cannot quantify. So we're looking at ways where we can quantify directly and rapidly by producing modified media. We're looking at ways, actually, for lack of better terms, to trick Campylobacter. Despite this abundance, we're still looking at a needle in a haystack. But with Campylobacter, you're looking at a needle in a haystack that's capable of moving around and doesn't want to be found, right?
Yeah.
So we're looking at ways to trick Campylobacter, actually, using its intrinsic ability to metabolize certain substrate in the gut of the chicken, where survival does not become preferential in that environment. The other thing is what we have mentioned previously, the use of antimicrobial light, the use of new phytochemicals that are safe for humans and safe to use in the chicken environment. One of the advantages of using these new techniques is that compounds like chlorine, despite being
Mhm.
their effectiveness in reducing pathogens, they have really bad reputation when it comes to consumers and even other markets, right?
Sure.
So maybe adopting these light techniques, these new phytochemical techniques can enhance the competitiveness of poultry as a protein and increase consumer confidence. So there are many things that we are looking in terms of biology. And of course, I always say my approach is before it comes to the processing factory, try to reduce it, because if you reduce it before at the farm, then you have an easier task while you're processing, right?
Mhm.
So we have also some host-specific interventions on the farm itself, and these range from the using of probiotic bacteria all the way to finding new molecules that can actually inhibit or trick Campylobacter inside the chicken host.
Listening to your comments there, particularly when you talked about some of the consumer perceptions of traditional intervention methods, I'm wondering if you see potential for the approaches you're looking at, say the lighting as an example, or some of the phytochemicals, to be applied in other areas of the food chain. Maybe it's monitoring water, environmental contamination. Do you think there are other practical applications beyond what we're talking about here, just specific to Campion processing?
Yeah, working with poultry, one thing that we have learned, for example, the recent rise in Campylobacter, many scientists believe is because of the effectiveness of controlling Salmonella,
Mm,
because we have focused on Salmonella that allowed for Campylobacter to emerge, right? So now way we think about stuff, okay, if it works for Campylobacter, does it work for other pathogens, like Salmonella, E. coli, or whatnot? the funding actually allowed us to test other things beyond Campylobacter. So we took those interventions and we tested them. Their effectiveness vary, but they are effective. So the whole premise is that the antimicrobial light and the phytochemicals, they have some effect on their own. If we combine the two of them, perhaps using lower concentrations of PAA and chlorine, will you sensitize these bacteria, whether it's Campylobacter, Salmonella, or others, to these interventions and vice versa? And what we have been seeing in the lab, yes, you do. So, yes, they can be applicable with other pathogens and they can be applicable with other foods also. So, if you're interested in something other than chicken meat, they can be used.
You always like when something you learn can be applied in a number of different practical ways. It's exciting. Dr. Kassem, as we wrap up our time together, if you had to summarize one message you want the industry to take away, whether it's producers, processors, your fellow poultry scientists, what do you want them to walk away with from your findings in this research?
As science evolves, new challenges will be discovered. And this is not necessarily something bad, because if you identify something when science evolves, then the solutions will evolve also. So we shouldn't be identifying of those strains. Now, because you identify those strains, you can target those strains because you know that they can be there, right? We shouldn't stagnate on how we look at food safety or how we look at processing of some of our favorite foods. And we shouldn't worry about trying something that can work. So don't just rely on what has been working throughout. And the other thing we should think about these pathogens, these are living organisms. They are going to evolve, they are going to change. And as they are changing, the challenges will change, right? So we have to be a little bit ahead. It's easier said than done, but you have some brilliant scientists and you have some brilliant people in the industry that are working on these things. And I think it can be done. It just needs a consensus and the will to tackle it.
Yeah, to quote the great philosopher, Dr. Ian Malcolm, life finds a way. So I appreciate that closing thought. Dr. Issmat Kassem with the University of Georgia, an assistant professor of microbiology focusing on antimicrobial resistance and infectious diseases. Really appreciate your insights, man. Thanks for joining us. Friends, if you enjoyed this conversation, it's a good time for you to subscribe to Unplucked wherever you listen to your favorite podcast. Give us a rating if you enjoyed the conversation. It helps others find the podcast. Of course, each week we bring you an expert like Dr. Kassem to talk about some of the most pressing issues in the poultry industry from a scientific angle. Check out our social media channels if you want to know what's happening in the life of the association. The Poultry Science Association just came from our inaugural Pacific Rim Scientific Conference in Macau, China. Dozens of fantastic abstracts presented there. Great number of really impactful symposia. And you can find out more from that meeting by following our social media channels at places like LinkedIn, Facebook, Instagram, and Threads. You can find us at all those various platforms. This episode of Unplugged was produced by Lindsay Johnston with editing and engineering by Michael Lunn of Parasol Video. It's going to do it for this week's episode. For the Poultry Science Association, I'm Andy Vance. See you next week.